So let me thank the organizers for a wonderful meeting. Yeah, can you hear me? Yeah. So thanks to all the organizers for this wonderful meeting. I'm going to talk about some fairly recent and pretty much unfinished work I've been doing with a couple of theorists, physicist colleagues in Copenhagen, but also influenced a lot by discussions with several experimental colleagues who've been working on a number of biological oscillators. Okay, so I mean, it's been at least 350 years, I think, since people have realized that when you take two oscillators and couple them, you can get phenomena like synchronization, entrainment, phase locking, and so on. And I'll try and use this one slide to summarize what we know. For instance, in a case where you have a single oscillator of the limit cycle type that Sagar was talking about, driven by an external periodic driver, okay? So this is a case where this is for a particular circle map, but many of these features apply pretty widely to any kind of limit cycle oscillator. So one of the things, the interesting things is that when you look at the driving frequency, which is on the x-axis, when that driving frequency is close to a rational multiple of the natural frequency of this limit cycle, you will find the possibility of entrained states, where the phases are locked, there's synchronization, and so on. When you are away from these rational multiples, that is, you're close to an irrational multiple, then you will find regions of unentrained states where the phases are slipping and moving and there's no particular relationship, okay? So when you start with very low amplitude, in the limit of this driving being very low amplitude, you're somewhere down here, and you find that the number of entrained states are very, very rare. Most of the time you're sort of in an unentrained state. But as you increase the amplitude of this driving, these regions of entrainment, they widen and they expand, and they're called Arnold tongues. And you start finding that the unentrained states start getting squeezed out until you reach some other point up here where the situation is reversed, and all the unentrained states, the irrational numbers, are sort of squeezed out and form a set of measure zero, whereas the entrained states sort of cover the whole space, okay? So it's not too bad an approximation, I think, for me to say that this region is very well understood. There is universality in the sense that pretty much any nonlinear oscillator will behave like this. We can answer pretty much any question you want to ask about this region. But this region is kind of interesting and not as well understood. What happens up in this region is that these Arnold tongues start overlapping. So you can get multiple entrained states, you can get more complex things as you go further up, you can get chaotic trajectories, and there are both experimental and some theoretical studies of this region. But in general, the behavior is more complicated. There's less universality in the sense that a lot of the details of the dynamics depends on the particular oscillator you're discussing, and so on. And there's many questions we don't understand about that region. So we've been sort of forced to confront some of these unanswered questions, because in the past few years, several of the biological oscillators we've been studying, where there is an important role of entrainment within the biological system, they turn out to be working in this region, okay? And I'll give you a few examples just to give you a sense. So here's an example of a particular oscillation that happens inside cells in your tail when you're an embryo. So this happens in pretty much every mammalian embryo. There are these cells which are oscillating, and the purpose of this oscillation ultimately is to convert this periodicity in time to a periodicity in space, which eventually forms your spine, your vertebrae, okay? And it turns out that there are these waves of oscillations and so on going on in this tail region, and the phase relationship between adjacent cells is very important and has to be very precisely maintained in order to get the right pattern, to get the right spacing of your vertebrae and so on. So that kind of phase relationship occurs because of the coupling between cells. And we know a fair amount of details about that molecularly now. We can estimate the kind of parameters of the coupling, and it very much works up in this state. Incidentally, I sort of said this was for a single oscillator driven by an external oscillator, the external period. But the same kind of picture more or less works for coupled oscillators, okay? In which case, this axis becomes not the amplitude of driving, but something like the strength of coupling. So when you look at this system, it's working very much up there. Another example comes from the group of Sanjay Sane and NCDS, where they've been studying the coupling between the wing oscillators, the wing beats in flies, and another structure that many of these flies has, which are called halteres. These also oscillate along with the wing, and they're sort of responsible. We don't know too much about how they work, but they are responsible in some way for helping the stabilization of flight. So if you cut these off, you know, the fly can fly, but it can't fly very stably. So we know, so these guys are able to sort of tune the frequency of the wing beat. And as they tune it and change the frequency of this and the oscillation of the haltere, which is driven by the wing, changes. And you can find entrained states, you can find unentrained states, and you can also see, for certain regions of parameters, an interesting hopping between different states. And this hopping is another feature of that region of overlapping Arnold tongues, and I'll come back to that. So a third example is, again, going back to oscillations inside cells. This is a particular protein that's very important for the immune system. And it oscillates in response to a large number of triggers, but amongst them things like inflammation that occurs when you, say, cut yourself or something, or when you have a wound. So in response to that, you see these very nice oscillations of these proteins going in and out of the nucleus of these cells with a time period of about an hour or so. And this, again, in the natural setting of a wound, often is driven by periodic sinusoidal or some kind of periodic variation of this trigger. So it is often driven by an external period. So actually, it's hard to study that kind of driving in vivo in a living cell, but in a living organism. But people have very recently, last year, actually managed to put these cells into a microfluidic chamber and subject them to oscillatory inputs of different frequencies, and they see what you would expect, some entrained and some non-entrained states, okay? So a few years before that, me and another colleague had been sort of looking at this kind of system. We had built a mathematical model for this oscillator and subjected it to an external periodic input and studied whether these Arnold tongues are there and so on. And of course, we find the Arnold tongues, and we can see that they also overlap and so on, and we can see all sorts of different kinds of behavior. For instance, I said that deep into the overlapping region, you could see chaotic trajectories. We can see that. More interestingly, close to the overlap, you can start seeing multiple different states of entrainment. Depending on these are deterministic simulations. Depending on the initial condition you start with, you can either fall into this mode or this mode, okay? But there are many questions we still don't fully understand, and we're trying to get a handle on. For instance, once you start overlapping, you are actually overlapping infinite Arnold tongues. Nevertheless, the number of stable attractors still seems to be finite, okay? But how many? Should there be 2 or 5? We see all sorts of things. Sometimes we see 2 like this, sometimes we see 3, 5, and so on. So we do not actually have an answer to the question of, even the simple question of just how many periodic attractors are actually stable, but we're getting a handle on that. Let me focus on another question that's sort of, again, inspired by the biological system, which is what is the effect of noise? In all these biological systems I've told you about, there is a lot of noise. In the system that I just told you about, this protein oscillations, for instance, the time period between these oscillations varies by about 30%. So you can imagine it's quite a lot, it's quite noisy. So of course, in the presence of noise, we do know, again, in the region where there's no overlap, in this low amplitude driving region, that noise generally shrinks the Arnold tongues. It causes them to sort of jump out into the unentrained states. And you will, so for a van der Pol, for instance, you'll find that the entrained states are much smaller. So these things all just shrink, okay? It turns out that in the overlapping region, it seems to be doing the opposite. It's actually expanding. And one of the things that's happening is that you start seeing hopping between these different entrained states, okay? So, of course, in simulations where we add noise, it's rather easy to see these different kinds of hopping between different multiple entrained states when you're in this region of overlapping tongues. But we wanted to go back and look at the experimental data. So we went back to this group and asked them if they see hopping. And here's a selection of some of the stuff they sent us. So you can, well, you know, it's, some of them are clear. There are a few cases, I think, like this one shows some interesting clear hopping between different entrained states. Some of them are not so clear and a bit noisy. In general, we need like larger, much longer time series to really be very clear about this, but there is some sign of hopping. So we're collaborating with these guys to get longer time series and see in general what kind of hopping we get, what kind of statistics we can see and so on. And that maybe I can report to you in the next meeting. But in the model, it's rather easy to, you know, run these simulations for a very, very long time. So we can, of course, see hopping of all sorts as we change driving frequencies and so on, and we can gather information about what's the probability of finding the system in a particular tongue, what's the time spent in particular tongues, or the time spent before you hop into another tongue. And I'll just mention one interesting thing that we see is that we see some very interesting memory effects. By memory effects, I simply mean in this context that a simple Markov, memoryless Markov process where you're jumping from one tongue to another with a certain rate does not describe the dynamics, at least in certain parameter regions. So we see that in certain situations, let's say you're in the 1:2 tongue, jumping from the 1:2 tongue to the 1:1 tongue, the probability of that may depend on, for instance, how you jumped into the 1:2 tongue in the first place. It may depend on how long you've been in the 1:2 tongue and things like that. Okay, so very briefly, let me just end by sort of describing to you some of the things we think are sort of underlying this kind of dynamics. It turns out that for this particular model, the geometry of these periodic attractors, these stable periodic attractors in this overlapping regime, we find has a very particular geometry of the sort of concentrically nested within each other. This is not just for the model I showed you, but we have models for these other couple of different biological oscillators. And all of them seem to show this kind of structure. And we think that this kind of particular geometric structure underlies a lot of the dynamical behavior. For instance, in the case of the memory effects, what seems to happen is that the probability of jumping, say, from one tongue to the other tongue is not uniform all around this trajectory. It's actually, say, higher in one place and lower in the other. And therefore, that is one of the things that underlies these memory effects, because you can get certain sequences of jumps which are much more probable than other sequences of jumps, depending on where you jump into and out of this trajectory. So, and we can explain some of the other things also with this kind of geometry. So let me leave you with some speculations. I've described to you a few situations where biological oscillators are coupled and show entrainment behavior, and the entrainment may actually play a very important role in the proper biological functioning of these systems. We think in general that all of these biological systems work in situations of pretty high noise, and that is a problem because you could easily jump into unentrained states. And I think what seems to be emerging, this is where I start to speculate, is that all of these biological oscillators deal with this problem of noise by pushing themselves into this overlapping regime, where instead of noise and unentrained states, you instead get these transitions and these hopping between the different modes, which might show interesting memory effects. And we may have stumbled upon a class of oscillators that seem to have a particular geometric structure that I think can explain many of these dynamical behaviors. So with that, I'll just stop and take questions. Thank you.
It's time for 1-2 questions.
Hello, I saw one of your slides, the coupled differential equation.
Yeah.
You kindly show me that slides again.
Oops. I think you meant this one?
Yeah, how you derive it actually.
How does it come? Well, yeah, so we know pretty much the molecular mechanisms that are involved in this oscillator. I've sort of drawn them very schematically here. There are things that transport certain molecules in and out of the cell. There are chemical sort of complex formation reactions where two proteins come together. essentially, we can write a model with all of these components, and then you can sort of simplify it to this because you know that certain processes are happening very fast, so you sort of average over them. Other processes are very slow compared to the time scales you're interested, so you ignore them. And if you do all that, then you can essentially boil down to what these dynamical variables are the concentrations of certain elements in this. Yeah.
Actually, you know how to plot or how to study the flow diagram of this coupling constant?
The what diagram?
Flow diagram, flow, F-L-O-W, flow.
I mean, yeah.
This coupling N, L, M, D-I-D-T. It's also appears in the RG equation in quantum theory. Those people are practicing quantum theory.
I'm not very aware of that. This kind of nonlinearity comes simply from a sort of a Michaelis-Menten-like nonlinearity, which comes from the formation of a complex. So it's basically chemical reactions converted to differential equations. But maybe we can talk more.
Okay, so I fear we have to move to the next speaker. Thank you again.